All right, awesome. So thank you, Eric and Sabira, for the kind invitation to come speak here. I'm so excited about this session, in part because I will be continuing continuing a lot of the themes you've already heard about in this morning's talks. So like Eric said, we've been doing a lot of things in my lab, and so I'll just give like a one-slide summary of all the crazy things that we've been doing. So we study a lot of sparse sensing control and biological engineered systems. So we work with a lot of this is done in invertebrate systems, and I have collaborators as well in quantifying natural behaviors, also mostly in invertebrate systems. I'm actually talking about that, which relates to Eric's talk about motor control in a different workshop tomorrow. I usually have a couple of people in my lab doing dynamical systems and control theory, just from a mathematical modeling perspective. Some of these folks don't even write papers in biology, so that's really fun as well. But I am talking about the last topic on the board here, which is neural decoding of brain-computer interfaces, mostly in humans, because that is a topic of today's lecture. Okay, so when I think about brain-computer interfacing in humans, I think about why we're doing it in the first place. And so this is what I came up with in terms of why we want BCIs in humans in the first place. The first goal is to assist individuals with physical and neurological disabilities. So Matt gave a really great highlight of that, one of those types of ways we can do this, to decode things that humans might want to do but are not able to do because of their physical or neurological disabilities. I can also think of ways to augment engineered systems with humans in the loops. So this is that we interact with a lot of digital devices in our lives. Usually the implementation is interfacing with your thumbs. You're typing on things, or you're looking at a cursor or something. But there might be possibilities of augmenting our interactions with the digital world by directly interfacing our brain activity with these engineered devices. And then lastly, I can think of ways to enhance your daily life in an increasingly data-rich world. We're all interfacing with lots of technology and lots of data all the time. And so you can kind of imagine a hypothetical future where we might be interfacing with data from the real world instead of through our sensory apparatus, but through our brain-computer interfacing directly. So I'm kind of curious because I asked my undergraduate class this question the other day. So I'm just going to ask this audience. This is a very different audience than an undergrad class, mostly pre-meds, right? If you could tomorrow, if it were possible to, and doesn't cost any money, and were ethically, you felt okay about it, would you get some kind of ECOG implant in your brain so that you can plug yourself into a computer? Raise your hand if you think you would do it.
Can I take it out if I want?
Can you take it out if you want to? Yeah, I think you can take it out if you want to. So you would do it? Wow, love it. Okay, so how many people are like, heck no, never? All right. This is fascinating. All right, I don't know where I come down on this, but I'm going to tell you the story of a set of projects that have been going on in my lab for a while. And it actually all started with my first PhD student, Nancy Wong, who is a computer science student. When she joined my lab, it was 2014, and this paper had just come out from University of Pittsburgh, where they demonstrated direct brain control of robotic arms. So I think this is probably not unfamiliar to this audience here. We've all seen this work. So Jan Schusterman has two different I think they're like 200 electrodes implanted in their motor cortex, and they were able to train decoders so that she was able to use this robotic arm to feed herself chocolate for the first time in a decade, which is really wonderful. And so Nancy and I were looking at this picture, and we're like, this is the coolest thing ever. We really want to do more of this. What can we do to contribute to this field so that we can make this better in the future? And so we were looking at this picture, and we recognized that there were a couple of things about this, as cool as it is, that could be better in the future. And the first one is, how do we take this outside the lab? Okay, so this works great as long as Nancy goes into the lab, but this is not really at that point feasible for using some kind of BCI outside the lab. So what do we have to do? What are the technologies and computational methods we have to develop so that it might be possible in the future for an individual like her to have this in her lab, in her kitchen, instead of in the laboratory? The second thing we identified was, how do we automate the graduate student? This was Nancy's primary directive, because she's a CSC grad student. She's always trying to okay, so how do we not have humans in here? And so we looked at it, it was like, wow, there's a gigantic research and clinical team in there with the participant every single day, fine-tuning the decoders, trying to make it work for them every single day. And so again, this is wonderful, this is great, super impressive work, but in the future, we might like to not have a research team with every individual, following them around every morning, recalibrating all of the decoders so that they could work for them. Okay, so that's the second goal. The third goal we came up with was, how do we have models that adapt to other people? Again, this is all wonderful for Jan, right? But it was a tremendous amount of calibration and retuning that was required so that this decoder would work for her. And it worked really well for her. But how would it be possible for us to not have to redo this for every single novel individual so that we can have this be more generalizable? So answering these 3 questions, or at least our attempt to answer these questions, starting from a series of papers started publishing in 2014, is what I'm going to talk to you about today. Okay, and it all starts with the same type of ECOG recordings that you've already heard about from Matt and from Uli as well. So these are participants who are in Harborview Hospital in Seattle. We worked with our neurosurgery collaborators, mostly with Jeff Ogeman and Andrew Koh at the time. They were the ones who did all the surgeries. And so these are human individuals who have intractable epilepsy, and so they're undergoing clinical monitoring for epilepsy, and they're implanted with approximately 100 surface intracortical electrodes, usually in one hemisphere. Sometimes we occasionally will get patients with different hemispheres implanted. And they're in the hospital doing this monitoring, and this is a Tuesday to Tuesday procedure, so they're there for a week. Okay? During this time, and this is what everyone else has been doing for a long time, a team of graduate students and postdocs come in and say, would you please participate in our research project? And of course, they're very kind, and they say yes. What we realized, what Nancy and I realized, was that there were actually continuous recordings 24/7, and they were throwing it away afterwards. This is 24/7 recordings of the individual doing whatever it is that they feel like doing in this hospital room. So they talk to their family, they eat, they sleep, they play on the computer, they watch TV, they read, they talk to their family members, and they talk to their clinicians. So they do all the things that you would do if you were in the hospital room like this one. So I call it naturalistic because it's not fully natural, right? They're not like outside skiing. This is not that type of natural behavior, but it's pretty natural in the context of things that we normally study in neuroscience. And so, of course, simultaneously, they have these recordings. I'll point out here that this is a bit different than some of the electrodes you heard from Matt and Uli. These are macro grids, and so the grids we have are usually 64 electrodes, so they're 8 by 8 grid. And the surgeons occasionally will tuck a few more different strips in there. The electrodes are quite large. So for context, the gray dots you see over there, they are 1 centimeter grid sizes. So they're pretty big dots, right? We're nowhere near Eric's single cell resolution, but we're sampling a large part of the cortical surface, but at very much coarser spatial resolution. That's the nature of the data that we have to analyze. Okay. So we've been collecting this data for a while, and usually I send a student down there with a storage device in their pocket because this is the hospital and they won't let us download the data. So I send a student down there on the bus and they come back. We now have a pretty large collection of data sets from over 50 patients so far. They're continuously monitored for at least 7 days each. Occasionally I'll stay for an extra week, so we have a couple of individuals where there's more like 2 weeks worth of data. For everyone, we have at least 64 electrodes, sometimes more like 100 electrodes. They're sampled at 1 kilohertz. Like I say, these are macro-grid ECOG, and so we're not getting single-cell resolution. It's more like average activity on the cortical surface between like 10,000-ish neurons or so, maybe more, okay? Importantly, at the same time, we have video of exactly what they are doing. During the day, it's RGB. At night, it's IR. And we also have audio monitoring. So I really want to talk to Matt about the audio stuff. We've tried, but I'm not talking about that today, in large part because we haven't found anything in it that's worth talking about yet. Okay, so what we did end up doing was really trying to lean into the neuroscience of naturalistic movements. So when you have these videos that are aimed at the individual during the hospital room, you can, they're not playing tennis, they're not skiing, right? So they're usually sitting in the hospital room. They usually have a blanket that goes up to their waist, so we can't really even see their lower limbs, but we have a pretty good view of their upper limbs. And so we can track their faces. We have a couple of things on facial recognition, and then we also can track the movements of their upper limbs. So we're really tracking the shoulder joints, the elbow joints, and the wrist joints as well. Given this type of data set, we started exploring. But we also, I'll just point out, I'm a big fan of the open science and open data publication movements. And so the first data set we published was actually back in 2017. We published the first agile data set, annotated joints in long-term ECOG. This was 4 participants at the time. A preview to the end of my talk, I'll tell you about the new data set that we just published last year, the Agile 12 data set, which is larger, bigger, and a lot nicer. And it's something that other folks have already written papers on. Thank you, Sabira, for using it. Okay, so one of the things we did was really try to pick out parts of the natural movements that are as comparable as possible to the type of experiments that other people have done with instructed movements. And so in instructed movements, the type of experiments you would do is that you would say something like, you tell the participant to like go pick up an object. So they'll go like and reach and pick up the object. And you get them to do that for as many times as you can get to repeat it, because they'll get mad at you if you do it too often. Okay? So here we're analyzing, we're trying to find opportunistically portions of the video where they happen to be doing that type of movement, but not because anyone told them to. They did it because they wanted to. Maybe they were reaching out to pick up an object. Maybe they were reaching out to eat food and bring it to their mouth. Maybe they're like me and they like talking with their hands a lot, and so they're just waving their hands a lot. Okay, so we're looking for movements that are repeatable and similar-ish to themselves, but is opportunistic, uninstructed, and they also happen at all times of during the day, so morning, afternoon, et cetera, and they happen in different social contexts. So sometimes they're alone, and sometimes they're with other individuals as well. And so we curated this pretty gigantic and my opinion, high-quality data set where you have joint ECOG data as well as behavioral tracking data of the upper limbs. So here is what you're about to see. This is just a small sample of the type of movements that we've been able to extract out of the data set. The participant here, as you can see, is sitting in their hospital room. I'm just going to play it. It's going to loop. So this is 16 movements where I've aligned all the videos to the initiation of the movement of the right arm, of the right wrist. So you can kind of see that in all of them, they're starting to move their right wrist. It's going to loop, so you can like look at each of them in turn. I will point out a couple of things that we thought to be particularly interesting in this data set. So first of all, is that those of you who have tried doing this markerless tracking thing, you feel my pain, okay? When the lighting changes that much, it is actually really difficult to get it to work this well. So you'll see here, they're in the hospital room, so sometimes it's IR video, sometimes it's RGB video, sometimes the window is open. I know it's Seattle, but sometimes the sun does actually come out outside, and it changes the lighting, and it makes the tracking really hard. Okay, so that's one thing. The other thing is you can see that sometimes they have a tray in front of them, so they're eating food, and sometimes they don't. Sometimes there is another individual. You can kind of see their feet in some of these videos. Occasionally it's a clinician, sometimes it's a family member. Like they have a little bed that's next to it, so I think they had a family member that was staying with them in some of these contexts. And you can also see, the other thing I'll point out is that we are lining it on the initiation of the movement of the right arm, because this is they had a left implant, so we're mostly tracking the contralateral arm, but we also had movements of both arms at the same time. So sometimes they only move one arm, sometimes they move both arms. And so there's a lot of behavioral variability already. Before we even get to the neural data, there's a lot of things that they're doing in terms of behavioral variability. And no one's really quantified this before in kind of like a naturalistic context and a spontaneous, uninstructed behavior. And that was the first thing we did, was just quantifying it. What is the variability of natural human arm movements? Gut check here, we have 12 subjects that we're analyzing. We're breaking it down by the movements and where they happen during the time of day. 12 is noon. As expected, they're more active during the day than they are at night. Some of them take a nap in the middle of the day, so that's what there's a little, like this bimodal distribution, when they're moving more in the morning and the afternoon. They do move in the middle of the night. This is the hospital, and so there's always noise and stuff. They don't actually sleep in a fully consolidated way like you might want to at home. Okay, so we can also quantify all of the different types of metadata in terms of like exactly what the movement is. These are not repeatable human or non-human primate movements, and so there's quite a bit of variability in terms of the reach duration, reach magnitude, reach angle, and onset speed. Some of these folks move really fast, and some of them are really quite slow. They're moving very gingerly and very carefully. And we also see that there tends to be, if you look at the reach angle column, there are more movements upwards than in any other direction, but we do see some movements downwards and then laterally as well. Well, the left and right movements were a little harder to find because I think they just didn't do it as much. There wasn't as much reason to reach to the left or the right. They're starting from kind of a resting position, kind of right here, and they're mostly reaching up. Sometimes they're scratching their heads, sometimes they're feeding themselves. So we see a lot of upwards movements more than other types of movements. We can also then quantify whether or not they are moving and talking at the same time. A couple of different things we made up. Bimanual ratio, a lot of these bimanual overlaps is whether or not they're moving one hand or both hands at the same time. Okay? So that's the behavior variability. And then we start digging into the neural variability. And so First piece of good news is that if you summarize all the data from these 12 individuals and collapse them onto the same coordinates by projecting them onto known cortical parcels, everything is extraordinarily consistent. This is exactly what we expect to see based on the previous results of controlled instructed movements. In particular, I'll point out that there is, these are spectrograms. In the middle, we have in red, the precentral region, and in green, the postcentral region. These are, again, exactly what you expect. We see this very characteristic increase in spectral power at the high frequency band and a corresponding decrease in power at the lower frequency band. So that's the red stripe and the blue stripe respectively in kind of like, human M1 and S1-ish areas. Okay, so far so good. There's almost nothing aligned to these movements in the temporal regions. Everything's good. This is like exactly what we expected. Okay, you ready? All right. And then we looked at all of them by themselves, and that's when everything broke down. And so the amount of individual variability here was actually astonishing to me. On average, everything was fine. And you see some individuals actually looked quite a bit like what you expect on average. So S7 here kind of has this very nice increasing power up here, decreasing power down here. Everything's good so far, right? S3 looks okay. But then there's individuals like S8 who are just missing the low frequency band altogether. It just isn't there. They don't have this thing. And then there's people like S12 where they do have an increase in the high frequency band, but it is shifted in the frequency band, right? It's not in the canonical high gamma range that had been described previously. And so accounting for this substantial individual variability was one of the core challenges we set out to solve in the beginning, right? We wanted to see, like, how do we actually build decoders that are possible to be useful for other people besides the one person that was participating in the experiment. And so contending with this variability was very much a goal of our studies, right? Like, what do we do with this? The fact that we acknowledge that your brain is different than mine. This is not an inconvenience. This is not a statistical anomaly. The difference between your brain and mine is the difference between you and me, and I value that difference in our individuality, right? So really embracing this variability in movements and also in neurovariability was something that I thought was both scientifically interesting, but also an interesting challenge in terms of building reliable decoders. So one thing we tried was a set of neural network decoders, and we started building on this framework called EEGNet, which is very popular, so many of you may have heard about it. It's a feed-forward convolutional neural net. It works. It has been shown to work really well for EEG data, and we started doing this for our ECOG data set as well. As always, the first thing we ran into was data quantity. Even though we had more data that you would have in a half-hour session, we had days and weeks of data, it was still not quite enough because these network architectures were pretty particularly power hungry. And so what we had to do was solve this challenge of data quantity by aggregating all of our participants. So this is a work led by a former postdoc, Steve Peterson, who now works at Facebook. And so what Steve decided to do was he decided to add two things on top of this EEG and build what we called an HDNet model architecture. So the first thing he did was actually project all of the individual brains onto a canonical brain, allowing us to fuse them together and actually use more data than we had for any individual participant. So we're training on all of the data at once. And the other thing he then added was a Hilbert transform. This is an intermediate layer in between the temporal convolution and the depthwise convolution in EEGNet. The reason we did this is because even though in theory a neural network can learn spectral features in the data, it doesn't have to. And so any expert knowledge we can inject into the network we observed was much better in terms of it converging onto a really good decoding solution more quickly. So the Hilbert transform was a way for us to inject in the knowledge from decades of human neuroscience knowledge that spectral features are actually, in fact, really, really good if you want to know something about the human electrographic record. And so we have a Hilbert transform in there, we have this weight matrix, and what we were able to do was using this network, we can then transfer a decoder of whether or not the person was about to move or not, so this is a binary classification task. We could do that with a new person sight on scene. Like I said, we have 12 participants, And so we did a variety of different generalizability tests. So within the same modality, we would train on 11 people and then test it on the 12th person. It worked pretty well. You can do this with a little more fine tuning. So with a little more fine tuning of just a subset of the layers on the 12th person, it does even better. In fact, if you do a little bit of fine tuning, and we're talking about like a dozen trials or so of fine tuning. So you train it on 11 people, you take that decoder, you fine tune it on a small handful of trials from the individual on that particular day, and it starts working about as well as a tailored decoder would. And we can also, and this is just a fun thing we did. Let me see if just like dug up with this other data set. This is not even our data set. It was a different data set from where it was an EEG recording of movements. And the model we trained on ECOG actually generalized to the EEG decoder. We had an EEG decoder without ever training one. It just worked, sight unseen. With a little more fine tuning, it worked even better. So here's a bit more quantitative results on the generalization and the transfer learning. What we could see is that HTNet outperformed other decoders when tested on unseen individuals and other other measurement modalities. And with a little bit of fine tuning, you can see that it starts achieving performance that works very, very well. So the thing that you want to compare here are kind of the pink bar versus the very last bar here, where you can see that the pink bar is kind of the bespoke decoder, and the blue one is the one we trained on everyone else and is slightly fine-tuned, and they start getting very close in terms of performance. But of course, all of this was based on a lot of training data, labeled data. Okay, so we all know this, and we all do it, right? Like over the years, I have hired a lot of undergrads to label a lot of frames of data to train not only the pose estimators, but also a lot of the ground truth data we're doing on this right? And so this is still like, this is not go towards the goal of having this be fully generalizable, having it be something that's actually useful to somebody for them to take home as something that's useful for them, because you still had to be able to like train all these decoders. And I can't tell you the number of undergrads that I paid to do this, and they only succeeded after a lot of hard work in labeling something like 2% of our data set, right? But that was what we needed to train the decoder. So this is not really good. This solution, especially when we're talking about clinical data, is simply not feasible. We can't do this. We can't have a scalable way of training these decoders without a different path. So I think Eva's talking later, and so I don't know, you'll mention some of your work in self-supervised learning as well, so thank you. Yeah, I don't know if you will, but maybe if you do. So she has some really cool work on a similar vein as well, trying to get around this idea of having labels. Our sort of very simplistic idea of getting around labels is doing decoders without labels with self-supervised learning. So this is an idea that we got from the ML literature. From the original paper we saw this was a cross-modal self-supervised decoding paper. And the idea there was, let's say you have you're analyzing multimodal YouTube videos, right? And you got stuff like you have a video stream and you have an audio stream, and you don't really know what's going on in these videos. But you do observe that there is temporal coincidence of things that are coherent around these modalities. So let's say I have an object, a video of an object, right? And it drops. You'll hear the sound of it hitting the table, and it's stopping the table at the same time. So by taking advantage of the fact that there is temporal coincidence of coherent structures between two different measurement modalities, and here it's the dropping of the object and the sound of it hitting the table, by learning those structures, I can then have those true streams of measurement modality actually train each other, right? It's kind of like a multimodal bootstrap clustering in some kind. Short version of it is that we did it. And we can do this not only for ECOG data and kinematics tracking data, we can do it for multiple data, more than 2 data streams as well, by doing this thing where we're basically randomly assigning cluster labels and then having the training labels, the targets, be from a different modality, and then swapping them randomly. And this thing eventually converges onto a solution that looks a heck of a lot like if you had trained a bespoke supervised decoder. Of course, full disclosure, this only works if there is some correlation in the first place, so you have to be really careful in terms of what data streams you're picking. But the one thing that we do know, especially in human biometric analysis, is that getting labels is hard, but getting long-term recordings actually kind of easy, right? We have tons of technologies and different measurable wearables and stuff. Right now, getting doing long-term longitudinal data for a long time is actually easier than getting labeled data. And so this is one way of doing so. So here I'm showing you one example of a data set we tried this on. This was 3 different data streams, and it was a 4-way classification task. And so the chance here is 25%. There's an EEG, 3D body position, as well as EMG of some of the calf muscles. And this is an individual kind of walking around in VR and also in real life on a treadmill. Okay? And so what I'm showing you here is the decoding accuracy trained on each of those modalities by themselves with supervised learning. So that's the blue bar. Here's what happens if you don't have the right answer. So this is essentially clustering, right? So this is like the ground. This is the straw man. This is the thing we're comparing to. We're trying to beat this because this is what happens if you essentially just do clustering. If you tie together more than one modality with this cross-modal self-supervised thing, it works better. If you use all 3, you get a decoder for the neural data. So this is an EEG decoder that works afterwards on just the EEG data. That works about as well as if you had done supervised decoding. So that's pretty cool, we thought. All right, cool. There's a lot more in this data set that we have managed to tune out. And so to support this effort, we spent a bunch of time working with the wonderful folks at NWB at Neural Data Without Borders, and released a Agile 12 data set that is currently on the internet. And so I'm just gonna make a plug here for this data set because there's so much more here that we cannot possibly analyze, and I would love it for this to be a resource for the community and for something that other people might find useful in your research project if you felt like using it. So the Agile 12 data set has the core data, which is a bunch of the ECOG recordings, as well as the tracked upper body poses. For obvious privacy reasons, I can't publish the raw videos themselves, but I can publish the tracked poses, okay? And then we have a ton of metadata. So we have events of these wrist movements that we've tracked. We also have, I did hire an undergrad, and he spent a long time coarsely labeling the behavior labels at about a 3-minute resolution. So there are some labels there of like, are they eating, are they talking, stuff like that. And then we have all of the data on the electrode features as well. The stuff is all in a standard data format in Neurodata Without Borders. To do this, we got the help of 2 individuals, Ben Dichter and Michael Sheed, who work for NWB, and they were wonderful, and they really helped us do this. I strongly recommend working with them because they were just so helpful because they have so much expertise. They made the whole conversion process a lot easier. The whole thing is deposited as a Dandy set on a public data repository Dandy. And in addition, we built a browser-based data dashboard. I know this is not an animation, but this is the thing that Ben built for us. And it makes it really easy to explore it without fully committing to download a few hundred gigabytes of data, right? So you don't have to do that to work with it at all. OK, so I've shown you here the structure of neural activity and executing naturalistic movements. We talked about how much variability there was in the behavior as well as the neural activity. And we talked about generalized decoding, how we can have these approaches to go towards the goal of decoding by taking advantage of lots of data from other people so that we can train it on the new person. We can also have an approach for decoding without labels. And importantly, we published the data set, as much of it as we could, as a resource to the community. Do I have a minute? No. OK. No. That's brutal. Thank you very much. And thank you all for listening.